Hi, Michelle Glass here. Welcome back to another episode of our Chapter 9 lecture series. We are here talking about examples of non-Mendelian inheritance. And the first pattern we want to talk about is what is called incomplete dominance. So in this case, we will see that when you have a heterozygous individual, you will have a phenotype that is intermediate between the homozygous dominant and the homozygous recessive. So we are going to see three possible phenotypes from this particular gene. So let's start by crossing a parental generation, a P generation, that's true breeding red flowers and white flowers. So we're looking here at snapdragons as our example. When you cross a true breeding red flower with a true breeding white flower, you will get a pink flower. So what's going on? How did red plus white equal pink? Okay, so let's do another cross. Let's take this heterozygous and cross it with another heterozygous. So we're going to cross two pink flowers. And when we do that, we get our F2 generation, which gives us 25% red flowers, 50% pink flowers, and 25% white flowers. So it's not blended. We haven't lost the red allele or the white allele. You can still get red flowers or white flowers when you cross pinks, but you also get pinks. And so this is our example of an intermediate dominant characteristic or trait. So what we're seeing is the heterozygous individual is going to have a phenotype that's in between the homozygous dominant and the homozygous recessive. So when we're looking at our pattern of incomplete dominance, you would expect your genotypic ratio to be 1 to 2 to 1, just like we've seen before. 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. But you're also going to see your phenotypic ratio is 1 to 2 to 1. It is the same. So we're going to have 1 with the dominant form, 2 with the intermediate trait, and 1 with the recessive form. So our signature of incomplete dominance is this same genotypic and phenotypic ratio for monohybrid crosses of heterozygous individuals. It will be 1 to 2 to 1 for both. Now, an example in humans is hypercholesterolemia. In this case, you have elevated blood cholesterol levels, which is causing some significant problems with your cardiovascular system and can lead to early death. This is controlled by a single gene, but it is an example of incomplete dominance. If you are homozygous dominant, then you have the healthy form, so you are able to remove cholesterol from your bloodstream and use it with your cells. Your cells will take it out of the bloodstream. If you are heterozygous, you are going to have 2 times the normal blood cholesterol levels. And if you are homozygous recessive, you're going to have 5 times the normal blood cholesterol levels. So clearly, the heterozygous individual is intermediate between, not as bad as the homozygous recessive, but also not as good as the homozygous dominant. Okay, that's it for now.